The sales of cocoa substrate to the professional horticultural industry in Europe started in 1989-1990. And since then, the product has been used as a substitute for potting soil as well as an alternative for rock wool. The use of cocoa as a growing substrate has considerably increased over the last few years. This is due to the high quality of the product and the environmentally friendly nature of the substrate. Cocoa substrate is made up of little grains, which are obtained from the shell of the nut of a coconut palm. It is in fact a byproduct from the cocoa fiber production. In 1862, the Royal Botanical Society introduced cocoa dust as a growing substrate. However, due to the rather poor quality, the substrate caused too many problems when used for short cultures. In 1994, a real breakthrough occurred as a special production process was developed, which facilitates the pre-treatment of cocoa. Canna is, without any doubt, a pioneer from the early days in the field of research. It is therefore not surprising that in 1995, Canna was the first company to market buffered cocoa that meets the RHP regulations. Cocoa has a number of superior qualities. Plants take root more intensely in cocoa dust and develop more fine root hairs than with other substrates. This is due to the fact that cocoa grains are made up of capillary microsponges, which can absorb as nearly as 1,000% of their own weight in water. This results in an enormous supply of water, air, and 100% absorbable nutrients, by which an improved crop development is facilitated. Another reason for better crop growth is the absence of diseases. Cocoa has its origins above ground and is therefore free of soil diseases. Next to this, it naturally contains the renowned Trichoderma mold. Thanks to the high content of lignin, it is possible to cultivate on Canna cocoa during a long period, without the risk of it settling or bedding down, and with maintenance of a nearly consistent air-water balance. Good quality cocoa can safely be reused several times. Potting soils often consist of peat, which has the disadvantage of settling down easily. Mixing the potting soil with 40% of cocoa results in a lighter mixture that will not settle anymore. Not all cocoa products being sold are suitable for the professional grower. Just as with potting soil, there are considerable differences in quality. Big differences can be found in the salt content, size and structure of the grains, used buffering and composting methods. A low salt content in the cocoa dust is essential for good crop development. The high-quality cocoa dust from Canna has been soaked in fresh water and is low in salt content. This in contrast with cocoa dust originating from other producing areas where seawater is used for soaking. In addition, Canna cocoa undergoes a composting process of at least 3 months. After it has been buffered and rinsed by way of a special treatment in the Netherlands, it meets the strict RHP regulations. Buffered cocoa gives the grower the advantage of starting with a neutral product. It is available in the form of slabs for use in Libra trays and in bags of 50 litres for pot cultures. Canna cocoa must be used in combination with Canna cocoa nutrition. This fertilizer has been specially developed and adapted to the buffered cocoa, and due to its special formula, it is suitable for the entire growth cycle. Canna cocoa contains also humic acids and fulvinic acids, 2 natural chelates, which guarantee an even better absorption of nutrients. A really user-friendly way of growing. But there are more developments: substrates with higher air content, improvements of production and growing methods. Canna's newest cocoa product, Coga, is a good example of this. The professional Coga boards are the result of the latest developments. The boards are made of coarse, textured pressed cocoa fibre. There are numerous advantages in using this substrate. It contains more air, is easier to control and better manageable in storage and transport. Therefore, it is perfect for the professional grower. 3 revolutionary fertilizers have been developed exclusively for the Coga boards: Coga Buffering Agent, Coga Vega and Coga Floris, which guarantee optimal conditions for the plant during root taking, growth and bloom. Growing on cocoa is not harmful to the bronchial tubes. The manufacture of cocoa involves no energy-consuming production processes. No natural resources are being exhausted, as cocoa nuts grow throughout the whole year, while peat, the basic ingredient of potting soil, needs 300 years to develop. Used cocoa does not create any waste disposal problems. It can be spread in the garden for soil melioration. Furthermore, the cocoa industry is a main source of revenue and contributes to the local economy. Canna cocoa is an easy medium to grow plants on. It is suitable to cultivate several successive crops on the same cocoa. In this film, we will show you just how easy it is. Cultivating in pots or on slabs is comparable. In this film, we show slabs. Make sure there are holes at the bottom of the slabs. These holes are necessary for a good drainage. Before you put the plants on the slab, you need to bring air into the slab. This is necessary to obtain an ideal air-water balance in the slab. Place the slab in the tray. Cut holes in the slabs on the spots where you will put plants. Push the cocoa in the plant holes a bit aside, so that the plants will stand firm in the cocoa from the start. Now the cutting is placed on the slab. It can be done standing in the cutting block, on a starter block, or in a Peter's pot. There are, of course, many other ways to place the cutting on the slab. As soon as the cuttings are placed on the slabs, it is recommended to use Rhizotonic because it stimulates root growth. Pour Rhizotonic and spray the leaves with it as well. Now it is time to fertilize the plants using Canna's uniquely formulated cocoa nutrients, Cocoa A and B. Start with an EC of 1 to 1.5. The plant is in the growing or vegetative phase. Increase the EC during the cultivation. Add Cannazine to stimulate bio-life in the slab and to achieve an optimal condition of the Canna cocoa. Check the pH and EC of the medium regularly by taking samples. As soon as you change to 12 hours of light a day, the plant gets a signal to flower. About 1 to 3 weeks later, the plant growth is tempered and it starts to bloom. Start adding PK 1314 4 weeks before harvesting. This results in big and firm crop development. These plants are ready to be harvested. It is very well possible to use Canna cocoa 3 times without any problems. If you reuse cocoa, we advise you to rinse the substrate with double doses of Cannazine after harvesting. Use of Cannazine during the growth minimizes the chance of passing diseases and helps old roots disappear. Let the rinse substrate rest for a day and you can start again. Pre-growing is a method with the purpose of obtaining broader and stronger plants without using space in the growing room. The small plants are planted out into a bigger pot or onto a bigger block before the plants are placed under light with a relatively blue spectrum, such as strip light or a metal halide light. There are several advantages using this method. You save space because you grow in a separate room. As a result of that, you save money as well. Pre-growing results in healthier, stronger plants that deliver a heavier harvest. Another way to pre-grow is in a Peter's pot. If you do not know what it is, then keep watching. A Peter's pot is a small pot of about 7 to 10 centimeters. Fill the Peter's pots with Canna cocoa. Put a rooted plant in every pot. Dip the pots in the Rhizotonic and spray the leaves as well. Rhizotonic contains useful minerals that are important for the root development. Put the plants under strip light or metal halide light. Leave the plants under this light for about a week. Now the plant is rooted well. Cut the bottom out of the pot. Put the plant with the pot on the growth medium. Repeat the pouring and spraying treatment with Rhizotonic. The plants start to root in the medium immediately. The plants are ready to start blooming now. This is what a fine result of pre-growing looks like. Reproducing by cutting is ideal if you are interested in cultivating your own favorite species. The plants will grow up evenly, you save money, and the risk that your plants get infections from outside are diminished. The most important condition for making good cuttings is a healthy mother plant. Put the starter plugs on a cutting tray and dip the tray in water. A tip: add trichoderma to the water. This stimulates root growth and provides protection from molds. Dip the tray with the starter plugs in the solution and wait until the plugs are saturated. Find a good shoot. Cut the shoot on the bias from the mother plant. This is your cutting. Remove the biggest leaves from the cutting. This prevents too much evaporation. Dip the cutting in the rooting powder or rooting gel. Wipe excess powder gel off because too much rooting powder has a negative effect on the root formation and plant growth. Use trichoderma powder as well to kill possible present germs. Squeeze excess water out of the starter plugs. If they are too wet, mold will be formed. Place the cuttings in the holes that you made for this purpose. At the beginning, the leaves of the cutting will be limp. It will disappear automatically. It is convenient to use a cutting box. If you don't have one, a plastic box covered with plastic film will do. Open the slide valves of the cutting box for ventilation before you place the cutting tray inside. Sufficient air is important for the plant. It prevents mold. Cuttings grow perfectly well under strip lighting. Make sure that the humidity in the box is minimal 75%. The cuttings will root best if the starter plugs are not too wet. Do not water the blocks for a couple of days. When the plugs get too dry, then dip the tray in water again. Be aware that the cuttings get irreversibly damaged if the starter plugs get too dry. After 7 to 10 days, the roots will be visible and the cuttings are ready to grow. In order to measure nutrition concentrations and acidity in peat, cocoa and soil, you have to dissolve the present minerals in water. The dilution factor that is used differs from country to country. In Holland, we use 1 part substrate in 1.5 parts of water. With the results, it is possible to take steps and adapt the nutrition pattern if necessary. In order to define the nutrition condition of the substrate, we start by taking a sample. The sample has to be a representative average of the root environment. Scoop a bit of substrate out of the root environment from as many as possible different spots. Put the gathered substrate in a bowl. To measure correctly, the samples must have the right humidity. The air pockets need to be saturated with water. If the substrate is too dry, it needs to be moisturized with demineralized water. It is moist enough if water runs out of the substrate when you squeeze it. Fill a measuring jug up to 150 milliliters with demineralized water. Add substrate up to 250 milliliters. Stir well and let the mixture rest for at least 2 hours. After that, the substance is ready to be filtered. Measure the filtrate. If the EC is lower than 0.6, then you need to increase the nutrition concentration in the nutrition tank. If the EC is higher than 1.2, then lower the nutrition concentration by adding water, or rinse with clean water. A good pH lies between 5.2 and 6. A pH that is too high or too low can be adjusted by altering the pH of the nutrition upwards or downwards with Canna pH adjusters.